Good morning, students. Let us continue medical terminology. Here I am mostly concentrating on lab related terminologies. That point you have to remember. Let us continue some of the few terminologies. See, we use a term like cancer, it's a general term. What is cancer? It is a disease caused when cell divide uncontrollably and spread into surrounding tissues. Here the word uncontrollably and spread into surrounding tissue, it is called cancer. Okay, it may be different, occurs in different part of the body. It is uncontrolled cell division. Okay, so what is tumor? Tumor term usually refers to cancer. Any cancerous growth is there, it is tumor. But there are two terms we have to remember. Tumor in general, it is a abnormal growth of body tissue that is often uncontrolled and progressive. So remember, tumor is not quite, it multiply or it spread, it invades or enter into other tissue or part of the body. So invasion is there. So tumor is a abnormal growth of body tissue that is often uncontrolled and progressive. Means it spread into other space, tissues, organs. It is not under control of body system. It is uncontrolled. Okay. Tumors usually two, benign and malignant. Tumor is the word we can use as malignant for tumor, but benign is a type of growth. It is a group of cell or it is a mass of compact mass of cells, and it remain at a site of origin without spreading into surrounding tissue. If it is spreads, it is cancerous. If it is unlimited growth, cell division, and it spreads to surrounding tissue, it invades into surrounding tissue, then it is called as that it is cancerous. Okay, infiltration we will call. It is give birth or metastasis. Metastasis means it give birth, it invade into other tissue. So benign cell grow as a compact mass and remain at the site of origin without spreading into surrounding tissue. Benign is, you can say, it is mild. It is encapsulated usually. Okay, most of the time it will not much harm. So without spreading. So what is malignant? Malignant means it's a tumor is made up of cancer cells and growth of the cell is uncontrolled. As I told, malignant is tumor, it is a cancerous. Malignant means tumor is cancerous, means tumor is made up of cancer cells. Okay, uncontrolled cell division is there. Okay, there is no control, and growth of because that much body ATP is utilized, our growth of the cell is uncontrolled and it gives spread to other tissue. Okay, part. So we call it as metastasis. So malignant tumor is always metastasis. It infiltrate into other part. Okay, it can easily spread. Cells can spread into surrounding tissue and spread into distant site. Okay, so if it is a lung cancer, it may spread to bone marrow. We don't know where it goes. Okay, so benign tumor, again I told, benign or benign tumor, the same word as a mass of cell growth that lack the ability to either invade neighboring tissues. So there is no metastasis in benign tumor. Usually we don't call it as tumor, benign growth. Okay, so what is mutagens? See, mutation is a term which changes in DNA, okay, sequence, okay, changes in the DNA which transfer to next generation. Okay, Mutagen is a agent that trigger changes in DNA sequence. It may be chemical agents, physical chemical agents. So agents that trigger changes in DNA sequence, leading to mutation. So this mutation passes to next generation. So oncogenes or carcinogens, agent capable of causing cancer in human. So oncogenes, they are chemicals or agents or substances capable of causing cancer in human. Also they are called as oncogenes or carcinogens, capable of causing aging. Some of the plastics you see in some of the chemicals like orthotolidine. There are many chemicals which cause cancer. Carcinogenic agent. So nicotine, whatever which cause cancer, called carcinogens, which is capable of causing cancer in human. So what is the torch test used in microbiology? It is a group of tests to screen multiple infection. TO refer to toxoplasma, toxoplasmosis caused by the organism Toxoplasma. Rubella R is a infection again. Cytomegalovirus virus. Then herpes caused by herpes simplex, and H is also refers to human immunodeficiency virus. So toxoplasma, rubella, cytomegalovirus, herpes simplex, and HIV. HIV is human immunodeficiency virus. This pack of test is called TORCH. What is PCR? You know already. Polymerase chain reaction is a technique utilized to amplify specific segment of DNA for a wide range of laboratory and clinical application. Now PCR becomes common to amplify, to get multiple copies, to amplify specific segment of DNA for a wide range of laboratory and clinical application. That is PCR. There are different types. So briefly, if you see RT-PCR, reverse transcriptase PCR. Here target is RNA instead of DNA. What is the meaning of screening test? So for example, screening test for diabetes. We may do random sugar or oral glucose control test, screening test. We do VDRL for syphilis, screening test. We do spot test for hepatitis. These are screening tests to perform as a preventive measures to detect the potential health problem or disease in someone in community that does not yet have signs and symptoms. Some are asymptomatic without symptoms, but that may be already spreaded into community, so our responsibility is to detect it soon. After spreading, once the symptoms started, it is difficult to control. So before that, if you know that something entered into the society, or you just want to screen some of the non-communicable diseases, whatever, before going into diagnostic, better to screen with the suspected age group and male, whatever. So just to know, this test which is performed as a preventive measure, to prevent, to detect a potential health problem or disease in someone that does not yet have symptoms. So like just above 40, we suspect type 2 diabetes, so better to screen with the random blood glucose any time in the day, or we give glucose and spot test, that is screening test. So HIV we can do tri-dot test, just to screen. If positive, we cannot confirm, we have to confirm with ELISA or Western blot. So but what is the diagnostic test? A type of test used to help diagnose a disease or a condition. So with that diagnostic test, we have to clinician have to treat the patient. So it is a diagnostic test. So for example, oral glucose tolerance test, OGTT, is a diagnostic test for diabetic mellitus. If it is above certain range, fasting is above 125 fasting blood sugar, it is diagnostic of diabetic mellitus. OGTT is diagnostic test for diabetes, but random glucose is not not all the times it is a diagnostic test. So okay, so some of the terms we use in the lab or hospital, there is ISO, International Organization for Standardization. ISO is International Organization for Standardization, anything standard quality control. It's an international committee of expert team to prepare and fulfill international standard procedure technique in medical laboratories as far medical lab is concerned. It is a general term, ISO, so to fulfill the requirement. If there is separate team, separate procedures, set of procedures we have to follow ISO standard. What is NABL? This particular term especially for lab. So National Accreditation Board for Testing and Calibration Laboratories. You can see the test report, RFTL, whatever. If lab is accredited with NABL, at the bottom there will be NABL accreditation seal will be there. So what is accreditation? Accreditation is an action or process of officially certifying. Here officially certifying is important, or being qualified or approved by an authorized body. It is not like internal peoples come from different field expert of same lab, means same different field. They may come from pathology, microbiology, cytology, histopathology, or a general expert, okay, specialist. So team, it is called team, body, to perform specific activities in an institution, laboratory, etc. So certifying, the action of definition for accreditation is action of process of officially certifying or being qualified or approved by an authorized body to perform specific activities. Okay, it should be accredited. So NABL is one of the accreditation body, National Accreditation Board for Testing and Calibration Laboratory. Okay, NABH is term same, but it is for hospital, National Accreditation Board for Hospital and other healthcare providers. Okay, so they have set of rules. You have to follow that one, whether inpatient, OP, ward, whatever, general staff, everything you have to follow that. So their team will come, they will inspect primary, secondary, there are different steps. So they recognize, finally they will certify, again they will come for follow up. There is a lot of procedure to get NABL and NABH accreditation. Okay, so CAP is international. Okay, the College of American Pathologists, it's a international accreditation body to certify quality control in clinical lab. After NABL we can go for CAP also. Okay, so what is the meaning of you seen screening test, diagnostic test? What is the meaning of gold standard test? It is the best available test for diagnosis and used for comparison. Okay, it is the best available test for diagnosis and used for comparison. I will give you one example. HIV we can do TRIDOT, just spot test we say. If you go blood bank, now they don't do like that. There is certain procedures. So generally we can say, or hepatitis we can do spot test. If it is positive, you have to confirm with ELISA, enzyme linked immunosorbent assay with the serum. Still it is positive, then we have to do Western blot for HIV. So for HIV confirmation is Western blot test, that is not the ELISA. So it is the gold standard test, is the best available test for diagnosis and is used for comparison. Okay, so these are some of the terminology in lab especially we use. Okay, thank you.